Okay. So, hmm. Good afternoon, everyone. So, today we'll be starting one discussion week 10 of the NPTEL course Biochemistry. And in this lecture, we'll be discussing mostly about the nitrogen metabolism and the urea cycle. Followed by, we'll start with the fatty acid oxidation. Then, let's start with the first question. Which of the following are incorrectly matched? So, what can be the correct answer? Okay. Okay, so the correct answer is B, C, D, F. That means these are incorrectly matched. Okay, and here is the explanation. So, this is the part of the nitrogen cycle that we see in nature. Okay, so basically, we have the nitrogen that needs to be converted to ammonia. Okay, so this nitrogen can come from the nitrate ion, and conversion of nitrate to nitrogen is denitrification, and then nitrogen to ammonia, it's nitrogen fixation, and it's basically done by rhizobium, Klebsiella, or azotobacter. Okay, then we have conversion from ammonia to nitrite is called as nitrification, and this is done by soil bacteria, and this is done mainly by the, for example, nitrosomonas. Okay, then conversion of nitrite to nitrate is done by nitrification only, because this is actually being oxidized to nitrate, and this is done by nitrobacter. Then conversion of nitrate to ammonia is also done by reduction by some anaerobic bacteria and mostly the plants. So this is basically the part of the nitrogen fixation. And then this ammonia is basically used for synthesis and degradation in plants and other microorganisms for amino acid and other reduced nitrogen carbon compounds. So now let's see the options. So we have azotobacter that converts atmospheric nitrogen to ammonia. Yes, this is correct. Then nitrosomonas converts nitrite in soil to nitrate. No, this is false. Because nitrosomonas, as we saw, it converts ammonia to nitrite in the soil. Then we have nitrobacter that converts ammonia in soil to nitrite. No. So nitrobacter basically converts nitrite to nitrate in the soil. So this is false. Then we have micrococcus denitrificans that converts nitrite in soil to nitrogen. No, this is false. Okay, this is denitrification. Okay, so it basically converts nitrate to nitrogen. Okay, so that's why this is false. Then we have Clibacilla that converts nitrogen to ammonia. So yes, that's correct. And then we have rhizobium that helps in nitrogen fixation in association with plants. Okay, so this is also correct. Okay, so out of these, B, C, D are correct, incorrectly matched, and F1 is the F1 was the correct answer. So it was by mistake written. So B, C, D and B, C, D are incorrectly matched. Okay. So any doubts in this question? Okay. So let's move on to the next question. Which of the following statements are true? So what can be the correct answer? Okay. Any more answers? B. Okay. Okay. So the correct answers are C and E. Okay. So let's see. The nitrogen fixing bacteria rhizobium are present in the nucleus of plant cells of the root nodules. No, this is false. It is present in the, so this is basically the diagram for the nitrogen fixation, and this is the bacteria. Okay, so it releases different factors and different molecules that help in nitrogen fixation. So this bacteria is generally found in the cytoplasm of the root nodules. Okay, so this is not present in the nucleus. So the first option is false. Then the reduction of nitrogen to ammonia is highly exergonic. That means it releases energy, okay, due to low activation energy of the reaction. No, this is false because it is highly exergonic because it requires high activation energy. Why it requires high activation energy? Because the nitrogen that we have, that is N triple bond N, that requires a delta G of around 900 kilojoule per mole. Okay, so this is sufficiently high amount of energy that is needed actually to break this point of this bond, this triple bond, and then it will convert to ammonia. Okay. So the next option, the activation energy of the reduction of nitrogen to ammonia can be attributed to the bond energy of the triple bond in dinitrogen. Yes, this is correct, because this activation energy is provided by the enzyme dinitrogenase. Okay, so this point is correct. Then ATP binding and hydrolysis at the dinitrogenase component of the nitrogenase complex provide the energy required to overcome the activation energy barrier. Yes, this is correct. Okay, because the nitrogenase, sorry, this is false, because the dinitrogenase, dinitrogen reductase enzyme component of the nitrogenase complex is actually responsible for ATP binding. Okay, not the dinitrogenase component. So this is false. Okay, then we have 8 electrons are directly involved in the reduction of nitrogen to ammonia. Yes, this is correct, and this is the equation that we see. So this is the overall equation that is being used for the completion of nitrogen reduction to ammonia. So this is nitrogen that combines with protons and 8 electrons followed by 16 ATP. So it forms two ammonium molecules that is actually be converted to ammonia, and by the release of 16 ADP and 16 Pi and the hydrogen molecule. Okay, is the explanation fine for this question? So the correct answer, that is the true statements out of the following, are C and E. So any doubts in this question? Okay, if not, then we'll move on to the next question. Which of the following are false? So what can be the correct answer? Okay, any more answers? Okay, so the correct answer is option B and D. Now let's discuss. So the nitrogenase complex consists of two enzymes, that is dinitrogen reductase and the dinitrogenase. So yes, this is correct. It has two components. One is the reductase part. So it has ATP binding site, and the dinitrogenase actually helps in conversion of nitrogen to ammonia. Okay. Then dinitrogenase reductase is a heterodimer consisting of four Fe4S redox centers. No, this is a homodimer. That means it has two identical peptide chains that have this 4Fe4S redox center. Okay, so option B is actually false. Then we have P clusters consist of two bridged pairs of 4S, 4Fe4S redox center. Yes, this is correct, and this is part of the dinitrogenase complex. Okay. Then we have overall nitrogenase. Overall, the nitrogenase complex is made up of eight polypeptide chains. No, this is false. So we have two polypeptide chains from the dinitrogenase reductase and four from the dinitrogenase. The total six polypeptide chains are involved in formation of the nitrogenase complex. Okay, so this is also false. Then we have one cycle of electron transfer from the reductase to dinitrogenase requires the hydrolysis of two ATP molecules. Yes, this is correct. Okay, in the last question we saw the equation. So for the reduction of one nitrogen molecule, we require 8 electrons and 16 ADP. Okay, so for one electron, we need two ATP molecules. Okay, so that's why this is correct. So the false options are option B and option D. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. Which of the following statements are true? So, what can be the correct answer? Okay, any more answers? So, okay, Any more answers? Okay, let's discuss. So the correct answer is B, C, D, E, and F. Okay, so let's see the statements. In all organisms, ammonia enters the amino acid metabolism through the glutamate synthase reaction. Okay, so this sorry, this glutamine synthetase reaction occurs in all organisms, in which glutamate is first made to react with gamma ATP to form gamma glutamyl phosphate, and this gamma glutamyl phosphate as an intermediate reacts with the ammonia to form glutamine. Okay, so this is the reaction that takes place for glutamine synthetase. Now, in bacteria and plants, we have another system that is glutamate synthase. Sorry. So this is the second reaction that takes place in bacteria and plants only, not in animals. In here, the alpha ketoglutarate and glutamine actually forms glutamate in the presence of NADPH. Okay, and then this ammonia is actually involved in like reaction with glutamate, alpha ketoglutarate, and this forms the L-glutamate out of this. Okay, so here the statement says in all organisms, the only pathway to for ammonia to enter the amino acid metabolism is glutamate synthetase. No, this happens only in plants and bacteria. Okay, and if it would be glutamine synthetase, then this statement would be correct. Okay, so the first statement is false. Then we have the glutamate synthase reaction is an example of transamination reaction. Yes, this is correct because here the amino group or the ammonia is transferred to alpha ketoglutarate that doesn't have an amino group. Okay, and this transfer of amino group to the alpha ketoglutarate forms glutamate, that is an amino acid. So yes, this is an example of transamination reaction. So this option is correct. Option B. Then beta glutamyl phosphate is formed as an intermediate in the glutamine synthetase reaction. Yes, this is also correct. Just now we discussed when glutamate reacts with ATP, it forms gamma glutamyl phosphate. So sorry, this would be gamma glutamyl. Okay, so gamma glutamyl phosphate, and then this gamma glutamyl phosphate reacts with ammonium group and it forms glutamine. Okay, so this option is also correct. Then we have D, that is glutamine synthetase is regulated by covalent modification through adenylation. So this is the second figure that shows is the regulation of the glutamine synthetase enzyme. So this glutamine synthetase is actually inactive when it reacts with AMP. So then what happens? The ATP that we have is actually transferred this adenylyl group to the glutamine synthetase, and this reaction is called as adenylation, and it tends to be inactive. Now, when this deadenylation happens, that means this adenylyl group is actually removed. That means now the glutamine synthetase is active right now. Okay, so this is option D is also correct. Then how this ATP is actually transferring this adenyl group to the glutamine synthetase, that is because of the protein, that is P2. Okay, and this P2 is actually under the regulation of uridylation. That means the transfer of uridine group to this P2. Okay, so this happens with the help of UTP. So if this UTP transfers its adenyl group, oh sorry, uridyl group to this P2, that means it is now inactive. It is now active and it can directly activate this ATP molecule to transfer this adenyl group to the glutamine synthetase. Now, when it's this uridyl group is removed, so now it can actually deadenylate it and it can activate this glutamine synthetase. Okay, so now the E point, the P2 protein associated with adenylyl transferase is regulated by the uridyl transferase mediated uridylation. Yes, just now we discussed. So E option is also correct. Then we have ATP and alpha ketoglutarate inhibit the deuridylation of P2. So this alpha ketoglutarate is and ATP, both these molecules inhibit the deuridylation. That means it allows the P2 to remain uridylate. Okay, so these green arrows actually suggest that it promotes the uridylation of this P2 molecule. So this F option is also correct. Then we have inorganic phosphate and glutamine promotes the uridylation of P2. So this inorganic phosphate and glutamine, no, both of these actually inhibits uridylation. Okay, so as you can see here, this cross marks means that both for inorganic phosphate and glutamine actually inhibits the uridylation of P2. So this is false. Then adenylation of glutamine synthetase occurs at threonine or serine residues. No, this is also false. This happens at tyrosine residues. Okay, so the true statements are B, C, D, E, and F. Okay, so any doubts in this question? Okay, if not, then we'll move on to the next question. So, which of the following reactions is not a part of the regulation of glutamine synthase? Actually, it would be synthetase. What can be the correct answer? Okay, any more answers? So, yes, the correct answer is option D. And just now we discussed that two things that helps in glutamine synthetase uh, regulation. First is the uridylation of the P2 protein and the adenylation of the glutamine synthetase. Okay, so the first option is uridylation of P2. Yes, this is involved. Then positive allosteric regulation by ATP on deuridylation of P2. Yes, this is also involved. Then we have adenylation of glutamine synthetase. Yes, this is also involved. And then positive allosteric regulation by tryptophan. No, This is not a part of the regulation of glutamine synthesis. So the correct answer is option D. Okay, so any doubts in this question? Okay, so let's move on to the next question. Which of the following statements are true? What can be the correct answer? Okay, C. Okay, anybody else? B, C, and D. Okay, okay. So the correct answer is B, C, and D. So the cell can store amino acids obtained from diet in the Golgi apparatus. No, this is false. Golgi apparatus is only used for transporting the proteins. Okay, so this is false. Then excess free amino acids in the cytoplasm can disrupt the osmotic balance. Yes, this is correct because they are present in the vacuoles in the cell. Okay, so that actually helps in maintaining the osmotic balance. So if the excess free amino acids are present, then it will disrupt the osmotic balance. The amino group from the amino acids is converted into ammonia in the liver. Yes, this is correct with the help of urea cycle. Okay. Then we have keto acid produced from the aspartic acid is used in gluconeogenesis. Yes, this is also correct. So what happens? The aspartic acid that is actually used in forming the keto acids, it enters Krebs cycle, and then from this it can directly be used to form glucose in gluconeogenesis. Okay, so this is also correct. Then the ammonia produced in the liver is combined with carbonic acid to form carbamoyl phosphate. No, this is also false. Okay. Then we have in humans, nitrogen is primarily excreted in the form of uric acid. No, uric not in uric acid, it's urea. Okay. So we primarily excrete urea, not uric acid. Okay. So the correct statements are option B, C, and D. Okay. So any doubts? Okay, if not, then we'll move on to the next question. Match the amino acids to their. Okay, wait. Uh, Yes, what is the doubt? So in this, the when there are, so for example, this is a cell, this is a nucleus. So what happens when the excess amino acids are produced in the body? So they are directly either stored in the form of like sorry, so they are directly directed to the cytoplasm, and there they actually are stored in the vacuoles. Okay, so now vacuoles are basically sites for where the storage happens, and it also maintains the osmotic balance. So when this amino acids are actually accumulated inside these vacuoles, so now the osmotic balance is actually disrupted. Okay, because the equilibrium between the formation and breakdown is disrupted. So that's why it leads to change in the osmotic balance. Okay, now it's clear.
Yes, sir.
Okay, so the next question is, match the amino acids to their corresponding keto acid produced by a transamination reaction. So now what you have to do is you have to match, and then you have to write the answer as an integer, in which, for example, for alanine it is oxaloacetate. So you have to write in this order in which you are matching. Okay. So what can be the correct answer? Okay. Anybody else? Okay. Okay, so the answer is 32145 So 32146 and 5. And this is the reaction. So alanine is actually formed from pyruvate. Okay, and so pyruvate is the keto acid for alanine. Then we have aspartate is actually formed from oxaloacetate. Okay, so this is the keto acid for aspartate. Then glutamate, we have alpha ketoglutarate. Okay, then for valine, we have alpha keto isovalerate. Then we have for phenylalanine, we have phenyl pyruvate, and for tyrosine, we have hydroxyphenyl pyruvate. Okay, so this you guys have to remember from which compound actually the amino acids are formed from the transamination reaction. Okay, so this is would be the correct order for the different amino acids that we can see here. Okay, so any doubts in this question? Okay, so let's move on to the next question. Which of the following statements are false? So what can be the correct answer? Okay. 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 So the correct answer is option A and E. Okay. So aminotransferases are functionally distinct from transaminases. No, both are the same enzymes. Okay, transaminases is the different name for the aminotransferase only. So this is false. The coenzyme involved in transamination reaction is derived from vitamin B6. Yes, this is paradoxal phosphate. Okay, so this is correct. The mechanism of transamination is a classic example of the ping pong mechanism. Yes, this is also correct. So, what happens in a ping pong mechanism? If this is the enzyme, so if this is the enzyme, and here the first substrate comes, it forms an intermediate, okay, and then it releases the product, that is P1. Now, because this enzyme has a molecule that actually is react with the second substrate. Okay, so now the second substrate comes and the final product is formed. Okay, and this enzyme is back. So this is the basic mechanism of the ping pong mechanism. So now what happens here? First the L amino acid comes. Sorry. So here first L amino acid comes. It actually modifies the enzyme and in the presence of the vitamin, so it forms a Schiff's base, that is L-damine. Okay, now this L-damine actually now converts to the quinoid intermediate. Okay, so they both can convert from each other and then it releases the keto acid, and here the pyridoxine phosphate is remaining. Now the alpha ketoglutarate comes and this amino group is transferred to alpha ketoglutarate forming glutamate. Okay, so this is a type of the ping pong mechanism. So this is also correct. Then aminotransferases involve pyridoxyl phosphate bound via an aldyne linkage. So yes, this is also correct. Just now we discussed that this aldyne is already formed when the L amino acid is actually con- joined to the enzyme. Okay. Then ammonia is released from the glycine in the hepatocyte mitochondria. No. Okay, ammonia is not released, it released in the form of urea. Okay, then excess amino acids in myocytes are ultimately converted into alanine, which enters the hepatocytes for further degradation. Yes, this is correct. So muscles basically the cells that form the muscles are myocytes. Okay, so these actually form alanine after the utilization of ammonia, and this alanine actually enters the liver via the bloodstream and it undergoes further degradation there. Okay, so this is also correct. So the false options are option E and option A. Okay, is the explanation fine? So actually the Schiff's base that is formed is the other name for the aldyne linkage. Okay, you can see here the Schiff's base intermediate or the aldyne that is formed. So that both are the same thing. Okay, so let's move on to the next question. Which of the following regarding transamination are true? So what can be the correct answer? Okay, any more answers? Okay, okay. So let's see. So the true statements are B, C, D, and E. Okay, and let's see. The aldyne form of the coenzyme is covalently linked to the enzyme at the leucine residue. No. it is the lysine residue. Okay, so this is false. Then aldyne form can undergo transamination. Yes, because it helps in the transferring of ammonium ion from the alpha ketoglutarate to the glutamate. So this is correct. Then we have aldyne form can undergo decarboxylation. Yes, this is also correct. So this aldyne form can actually lead to the removal of this carboxyl group also. Okay. Then a carboanion form intermediate is formed during the reaction. Yes, this is also correct. And it is converted to the quinoid intermediate. So both the intermediates are formed during the reaction. So both the statements are correct. The reaction involves one substrate and produces two products. No. Two substrates are there. First is the amino acid. Second is alpha ketoglutarate. Okay. And the products are also two. One is the keto acid and the second one is glutamate. So two substrates and two products. Okay, so the true statements are B, C, D, and E. Okay, so is the explanation fine? Okay, so let's move on to the next question. An in silico study identified drug X has the potential to bind and phosphorylate certain proteins. The phosphorylated forms of these proteins stimulate lipid breakdown. Based on this, which of the following are possibly phosphorylated by drug X? So, what can be the correct answer? Okay. Okay, so the correct answer is option E. That means all of the above proteins are actually involved in, if the drug X is actually being used, so all the proteins will be phosphorylated. Okay, and here is the regulation of the fatty acid metabolism. So what happens? There is a hormone that have a receptor, so it this receptor actually activates the adenylyl cyclase. So this means that it phosphorylate adenylyl cyclase. Now this adenylyl cyclase actually converts ATP to cyclic AMP, that is the secondary messenger. This cyclic AMP activates protein kinase A, so that is actually being phosphorylated. And then this protein kinase A, as it's a kinase, it will phosphorylate different proteins. So one is perilipin, and the other is the hormone sensitive lipase. Okay, now both of these will help this lipid droplet to get converted from triacylglycerol to fatty acid. Then this fatty acid will enter the bloodstream and it will reach the myocytes, okay, or liver via the fatty acid transporter, and where the beta oxidation helps in forming the carbon dioxide and the ATP from these free fatty acids. Okay, so this is the basic mechanism where it helps. Now, the different proteins that are involved in this are adenylyl cyclase, then we have protein kinase A, perilipin, and the hormone sensitive lipase. All of them are actually phosphorylated in the fatty acid oxidation. So now get back to the question. So an in silico study identified drug X that have a potential to bind and phosphorylate certain proteins. The phosphorylated form of these proteins stimulate lipid breakdown. Based on this, which of the following possibly phosphorylated by drug X? Okay, so all of these proteins are actually involved in degradation of lipids. Okay, so that's why all of these proteins are actually being phosphorylated by the drug X. So the correct answer is option E, that is all of these. Okay, is the explanation fine? Okay, so let's move on to the next question. Observe the diagram given below and identify the correct statements. So this is a diagram given from the urea cycle to identify the correct statement. Okay, A and D. Okay, so the correct answer are B, C, D, and here is the urea cycle. Okay, so let's see. So first, what happens? This ammonia group combines with the carbon dioxide and phosphate to form the carbamyl phosphate. This carbamyl phosphate reacts with ornithine and it releases citrulline. Okay, so this A is actually carbamyl phosphate and B is ornithine. Okay, then it forms citrulline in the mitochondrial matrix. After this, it is released into the cytoplasm and it combines with ATP to form a citrulyl AMP intermediate, and then comes the aspartate. So this C is actually aspartate that combines and forms the arginosuccinate. Okay, then this arginosuccinate releases arginine and fumarate. So this D is actually fumarate. Okay, so now this arginine is again converted to ornithine by the release of urea. So that's why this is called as a urea cycle. Now we'll see the options. So in a cell, compound A is the end product of a reaction between ammonia and bicarbonate. So compound A, that is carbamyl phosphate, is the end reaction of ammonia and bicarbonate. No, okay, because here we will require phosphate also. Then we have the compound B and lysine differ by one CH2 unit. So compound B was ornithine, and here it will differ only by the one CH2 unit. Okay, so here is the structure of ornithine, and this is the lysine structure. And here we'll attach only one CH2 and it will form lysine. Okay, so this is correct. Then we have the side chain of compound C has the lowest pKr value among all the 20 canonical amino acids. So compound C is aspartate and the side chain of aspartate is CH2COOH. So the pKa value of this is the lowest among the side chains of the amino acids. So yes, this is correct. Then we have option D. The compound D is the trans isomer of malic acid. So compound D is fumarate. So yes, malic acid is the cis form and fumarate is the trans form of the same compound. Okay, so this is also correct. Then all precursors of the given cycle are present within the cytosol of the same cell. No, citrulline and citrulline is actually and the carbamyl phosphate are present in the mitochondrial matrix. Rest of the intermediates are present in the cytosol. So this is also false. So the correct statements are B, C, and D. Okay. So any doubts in this question? Okay. So let's move on to the next question. Which of the following are true? What can be the correct answer? Okay. Any more answers? Okay, so the correct answer is B and D. So let's see. Just hydrolysis of ATP is sufficient to counter the high activation energy of nitrogen reduction? No. Okay, that's why the enzyme nitrogenase complex is actually needed to counteract this high activation energy of the dinitrogen. Okay, so this is false. Triacyl glycerols have no effect on cytosolic osmolarity. Yes, because triacyl glycerols are large molecules. If those are broken down to free fatty acids, now these can actually pose a difference or effect on the cell osmolarity. So option B is correct. The role of apolipoproteins is to hold the triacyl glycerol place in the chylomicrons. No. Okay. Then ammonia has 2 points of entry into the urea cycle. Yes, this is correct. So first is from the carbamoyl phosphate, and the second one is, so first is the carbamyl phosphate and the second one is the aspartate. So in the form of aspartate also the ammonia can enter the urea cycle. Okay, so th- yes, this is correct. So in the form of aspartate. Okay, so options B and D are correct. Okay, so any doubts in this question? Okay, so let's move on to the next question. Consider that 1 gram of triacylglycerols can store 40 kilojoule of energy. If 20% of the body mass of adult weighing 100 kg consists of triacylglycerols, what is the total available fuel reserve? So what can be the correct answer? 800. Okay. Okay, so let's solve. So if a person has 100 kg mass and out of those 20% is triacylglycerol, that means what will be the mass of the triacylglycerol? It would be 20 kgs. Okay. Then for 1 gram of triacylglycerols, 40 kilojoule of energy is stored. And for 20 kg, it would be 40 into 20 into 1000. Okay. So because it was in kg, so I have to convert it into grams. So 40 into 20 into 1000. This much kilojoule of energy is actually being stored. So this would be 8 into 10 to the power 5 kilojoule of energy is actually stored. Okay. So the integer value that we'll put here is 8. Okay. Is the explanation fine? Okay. So let's move on to the next question. This is in continuation with the last one. If a person in above question needs 10,000 kilojoule of energy per day to survive, how long can she survive off just the fuel reserve from the fatty acid oxidation? So, what can be the correct answer? Okay. So, yes, the fuel reserve that was stored from the triacylglycerol was 8 into 10 to the power 5 kilojoule, and the person needs 10,000 kilojoules per day to survive. Okay, so that means we'll give 80 days. So this much amount of energy she has, and this much is needed per day. So from only beta oxidation of fatty acid, she can directly survive for 80 days. Okay, so the correct answer is 80 days. So any doubts in this question? Okay, so let's move on to the next one. Which of the following processes will be affected if bile salts are not produced? So, what can be the correct answer? Okay, so yes, the correct answer is option B. And here is the answer. So what happens? These bile salts are actually released from the these emulsified dietary fats in the small intestine, forming micelles. Okay, so they are released from the gallbladder and the liver in combination. So both of them release it into the intestine, small intestine, and then followed by the intestinal lipase, and they degrade triacylglycerol, and then fatty acids are broken down in the intestinal mucosa. Okay, so now they move as a chylomicron, and then this chylomicron move through the lymphatic system and the bloodstream in tissues, and this lipoprotein lipase activated by the apoC2 in the capillaries that converts triacylglycerol to fatty acid and glycerol. Then this free fatty acid entered the cells, and these are the myocytes or the adipose tissues. So this is the basic physiological pathway of degradation of the fats. Okay, so now let's see. So these bile salts are actually being important in the digestion in the small intestine. Okay, so these are directly released from the liver to the small intestine. So lipid uptake in the intestine will be affected if the bile salts are not produced. Okay, stomach, for stomach, it will not be have any effect because the stomach does not require the bile salts for degradation. Okay, so it will be only the intestine that will be affected, and only the lipid metabolism, not the starch metabolism, that will be affected. Okay, so this is the correct answer, option B. So any doubts in this question? Okay, so let's move on to the next one. Researchers transferred a gene from the soybean plant to a different legume plant called Lotus cornicellatus. They allowed the bacterium Rhizobium loti to infect the plant and form root nodules. They found that the gene that they had transferred and expressed only in the root nodules containing Rhizobium loti. What protein might this gene encode? So, what can be the correct answer? Okay, yes, the correct answer is option C. So, what happens? Rhizobium is only needed for the infection, and the nitrogenase complex is actually present in the rhizobium. Okay, so this all nitrogenase complex is contributed by the rhizobium. Only the basic need is the depletion of oxygen, and that is done by leg hemoglobin. Okay, and this leg hemoglobin is spread from the plant. So, leguminous plants have this leg hemoglobin, and in this question, the other things are fine. Only the gene that is expressed in the root nodule specifically is the leg hemoglobin. Okay, so because it's important for the rhizobium to actually reduce the nitrogen to ammonia. Okay, so any doubts in this question? Okay, so let's move on to the next one. Shown below is the image in the is the molecule gamma aminobutyric acid, that is GABA. Which amino acid in this molecule a derivative of, and what function is it known to perform? So what can be the correct answer? So the correct answer is option A. And so glutamate is actually converted to GABA, that is gamma aminobutyric acid, by the presence of the enzyme glutamate decarboxylase. Okay, so this carboxyl group, sorry, this one is actually removed in the form of CO2, and this neurotransmitter is actually formed. So this is a inhibitory neurotransmitter that is formed from glutamic acid, so that it helps in neurotransmission basically. Okay, so any doubts in this question? Okay, so let's move on to the next question. Which of the following statements are true? So what can be the correct answer? D also. Okay. So the correct answer is A, C, and D. So the biological role of fatty acid oxidation can differ among organs and organisms. Yes, this is correct, and this is based upon the amount of energy that is needed for the organism or the organ. If more amount of energy is needed, so the fatty acids are also oxidized more in that case. Okay, then we have the chylomicrons are apolipoproteins. No, this is false. Then carnitine deficiency can result in a reduced rate of oleate oxidation. Yes, this is correct. Okay, because carnitine is actually a transporter for the fatty acids. Okay, so it transports it to the matrix. So if carnitine is deficient, then it's the reduced rate of oleate oxidation. Okay, then we have carnitine deficiency can result in muscle weakness and muscle cramps. Yes, this is also correct, because if carnitine is deficient in the body, that means there will be less transport of fatty acids to the matrix of mitochondria. So if that is the case, so what will happen? There will be less beta oxidation, and then leading to muscle fatigue and weakness. Okay, so the correct statements are option A, C, and D. Okay, so any doubts in this question? If not, then we'll move on to the next question. Which of these kind of molecules, when present in excess amounts, can be stored in the body for future use? So, what can be the correct answer? Okay, B and C. Okay, so the correct answer is option B and D. So, yes, lipids can actually be stored for long term use in the form of triacylglycerols, that is the storage component. The nucleotides can also be stored. Okay, so sorry, if not needed, they are actually being stored. Yes, in carbohydrates, we have glycogen that can actually be stored for long term use. Okay, so option C will also be included here. Amino acids we generally don't store because these are derived from these carbohydrates only. So all these three will be stored in the body for future use. Okay, so the correct answer is B, C, and D. So nucleotides are basically when they are not in use. So generally nucleotides are not used for energy source as an energy source, but when for basically replication and the different nucleotide metabolism, in that case nucleotides can easily be stored for long term use. Okay, so this was the last question for today. So if you guys have any doubt in the presentation or in this week, you can ask. Okay, if not, then we'll close the session for today. Thank you guys for joining in. Yes, sometimes. Okay, thank you.